All right. Well, welcome everyone. My name is Charlie Doobie. I'm the head of Regenerative Partnerships here at Agrology. Really excited about this webinar. Joined by my two colleagues and good friends. I'll let them introduce themselves in a second. But yeah, we're really excited to have everyone. The idea of this webinar is we had a whole year of really interesting, amazing data. So we're going to dive in and look at some of the patterns that emerged from different trials, look at some insights that popped up for us, some questions that are remaining and that we're going to be looking into next year. And yeah, overall, just take a deep dive into the ground truth data that Agrology gathered. So Jay, I'll hand it off to you, and then we'll move on to Dana.
Yeah, I'm Jay. I'm head of agribusiness development here. So work with Charlie on all of our sales and partnerships. Yeah, really excited. I think this webinar format is a bit different. Usually we have like an industry leader or customer on, but today it's just Team Agrology, which is going to allow us to cover a lot of ground and look at a lot of different trials. And I think that's going to be really fun because we get to share sort of a lot of ideas with our audience today.
Great. Thanks, Jay. My name is Dana Ravallo. I'm the head of customer success at Agrology. I provide data to our growers, onboard them onto the grower portal, make sure they're able to access the data that we're collecting, and provide them with actionable insights on the trials that they're running.
Awesome. So, with no further ado, we'll jump into some of the trials and some of the data we gathered this year. Before we start that, just some quick housekeeping. Please keep yourself on mute. We have a chat open, and please add any and all questions into the chat. We'll probably try to answer most of them live. If we don't get to them, then we'll circle back at the end. But yeah, we're excited to answer any and all questions. If we don't have the answer to the question, then we'll try to track it down or figure it out. But yeah, in our data, there are definitely some places where there's more questions than answers, and that's a really exciting piece of this. Everyone will receive a recording. I'll send up a follow-up email, so no need to be furiously taking notes. And also, a really important note, we at Agrology take our customer data privacy very, very seriously. Customers own their data entirely, and so we got permission from all the customers whose data we'll be sharing today. Some of them asked to be anonymized, so that'll be just anonymous data, but some of them are okay with us sharing their name and kind of giving them credit where credit's due. So yeah, with that, we will jump right in. So just in case, you're unaware or kind of unfamiliar with the agrology system, I'm just going to give a quick kind of overview of our arbiter system. We have two systems at agrology. One is the Sentinel, which does all sorts of climate monitoring, looks at soil moisture, soil temperature. It has like a digital nose, so we can look at VOCs and the risk of smoke taint risk in vineyards. But mostly, we're going to be looking at the data from our arbiter system today. The Arbiter system is a really amazing, unique system. Here on the top kind of left corner, we have kind of a diagram of it. It looks at CO2 concentration in three different places. So it looks at it through a soil chamber, looking at what's called soil respiration. If anyone's ever done a Haney soil test or like a Solvita burst test, this is the same kind of approach in technology. And essentially, the microbes in the soil, as they go through their lives and they are decomposing organic matter and they're cycling nutrients, they breathe out CO2. And so the total amount of CO2 that's coming out of the soil is the total amount of microbial activity there. During a Haney test, they'll actually take the soil, you ship it into a lab, they dry it all the way down, and then they re-moisten it, and then they get the total parts per million of CO2. And then based on that, they can tell you how much microbial life there is in your soil. We do the same thing, but in real world conditions. So not in laboratory conditions or sort of artificial conditions. We're telling you, instead of what the potential activity of the soil could be, we're telling you what the actual activity is based on the real world conditions of soil moisture and the climate that you're in and the practices that you're doing. So it's this really interesting continuous measurement of the amount of life in your soil. And you all probably are familiar, but I'll just give a quick rundown of why soil life matters so much. Soil life, in my opinion, is the foundation of the pyramid of a successful farm, really. It's the base of the pyramid of ecological function in the soil. The more soil activity that we have, we're seeing that biology is the main thing that's actually creating soil structure. That's because bacteria, as well as root exudates, creates this kind of amazing mucus that binds soil particles together. And then those small aggregates are bound together by fungal hyphae into larger macro aggregates. This is what creates soil structure. This is what creates pores. Why are soil pores important? Well, number one, we all know that compaction does really bad things for crop performance. But when you have those soil pores, it allows water to infiltrate easily. And then you also get the right mix of oxygen and water so that crops can really perform. Soil biology is also responsible for nutrient cycling, for carbon cycling. And kind of to jump back, that is really what we're monitoring with the arbiter system. We're monitoring the carbon cycle. So we're not just looking at the kind of point in time, this is how much TOC you have, total organic carbon. We're looking at the carbon cycle, and we're looking at how active and kind of vital and vibrant that cycle is. But microbes are also responsible for cycling nitrogen and kind of all of the other macro nutrients. We also know that when there's a very healthy, biodiverse, vital soil microbiome, we see better pest, disease, and stress resistance in crops. That is because there's this really magical thing that happens right at the rhizosphere. So at the level of the root hairs and the roots, there's this exchange of energy and nutrients. And the more kind of the larger and more robust that dialogue is between soil microbiome and plant roots, the better a plant can produce these biochemicals that are called secondary plant metabolites. Secondary plant metabolites, there's lots of examples of them, like in wine, it's like the polyphenol compounds and the tannins. Nicotine in tobacco plants is a great example of it. And nicotine is a really interesting example because that shows, probably a lot of folks know, if you plant tobacco on the edges of your fields, pests really don't like it, right? Nicotine's a great example of that type of secondary plant metabolite, that it is a very active and very strong pest deterrent. And then, of course, the one that we're kind of most familiar with, which is increased carbon sequestration. When we have soil life, that is a precursor to carbon sequestration. Carbon comes into the soil through the liquid carbon pathway. It's absorbed via photosynthesis. For a lot of plants, up to 50% of the energy that they gather from the sun is released into the soil microbiome. And then it is slowly processed by the soil microbiome and humified, turned into humus, which is that stable carbon form that we're really looking for to not only fight climate change, but also it's really good for crop production. So long story short, Agrology monitors that amount of microbial activity through soil respiration. And that's a lot of the slides and the data that we'll be sharing today, is that real-time continuous monitoring of respiration. So with that, I will hand it off to talk about this trial we did looking at different types of tillage and its effect on the microbial communities and their function.
Great. Thanks, Charlie, for that comprehensive overview on soil health. So this is a tillage trial we did at Redwood Empire Vineyard Management, and this tillage trial provides insights into the effects of different tillage methods on soil health, water retention, and overall productivity. If you look, the blue line on this graph is the no-till line. We have a yellow line that is also no-till, but on a lower vigor block in that vineyard. The green line is every other row was tilled down the middle, and then the red line was the full tillage. So this trial highlights balancing soil compaction, water infiltration, microbial health. Full till, often necessary for weed control and more of a traditional way of farming, um, provides good soil preparation, but disrupts the soil structure, leading to faster drying and reduced microbial activity. On the other hand, the no-till preserves soil structure, which is beneficial for microbial life, water retention, reducing the need for irrigation and improving overall soil health. The every other row till provides a middle ground, offering some benefits of no-till while still allowing for easier management. The no-till methods, while potentially more labor intensive and difficult to implement to transition, offer longer term benefits like improved water retention, like we'll see on the next slide, reduced evaporation, and better microbial health. If you see the blue line, the no-till actually very closely mirrors the full till line, showing that there's a very similar amount of respiration. Um, what's very impressive about this is that this was the very first year that they implemented this trial. Um, so the blue line, by tilling, you're also incorporating a lot of oxygen, burning off a lot of the microbial life, while the no-till line is showing the increased respiration of the soil life in the soil, a lot of the root exudates and
Yeah.
everything else. So overall, this was a very successful trial for the first year.
Yeah, I
Yeah.
think one thing that was interesting that I like to look at on this one is how all the lines are pretty close together at the beginning of the year, and then they kind of slowly start to diverge. And like you said, Dana, we oftentimes see in tillage that there is a huge amount of respiration, but it kind of burns off all the carbon. So you see, I like to use the metaphor of like it's like throwing really small kindling or logs into a fire. Like it burns off really quickly. You get this huge amount of activity, huge amount of heat, but you don't get that long sustained microbial activity. And oftentimes we see it crash either later that year or the next year. We know that consistent tillage does very bad things for the soil microbiome. Number one, by just disrupting all their homes and kind of slicing up the structure and destroying that pore space. But also, you're creating like this massive feast. You're chopping up all of the soil organic matter, and then you are adding all this oxygen. So there's this massive kind of burst of activity, but then all that food is gone for later in the season. All the microbes have gone to town and had this huge sort of feast, but then there's nothing left to
Yeah.
kind of keep that activity.
I think the remarkable thing about this being the first year that they're sort of switching to these practices is how the no-till block is keeping up in terms of microbial activity, right? We typically see a huge burst associated with tillage, but you can see that no-till block keeps up. And we're really excited about continuing this trial and starting to see those lines start to tease apart. Um, yeah, and I know that the low vigor line here in this tillage trial, it's a more sandy soil, lot less organic matter. Um, so, we expect that to sort of lag behind the others. Yeah.
Okay, next slide here.
Yeah, and so here's the other metric that we were gauging this trial on is the water holding capacity of the soil. So again, just like the last slide, the blue line is the no-till, the yellow line is the lower vigor, the green line was every other row tilled, and the red line is the full tillage. So we're looking at the water holding capacity and of the soil moisture from April through July in Sonoma County. So the results clearly show the importance of tillage practices on water holding capacity. No-till practices yielded the highest water retention levels, allowing the soil moisture to be more effectively held rather than full till or the every other row till. The disruption of the soil structure in the full till and resulted in a quicker drying pattern. These findings really emphasize the link between soil structure and moisture availability, especially in drier regions where we're not getting any rain in Sonoma County. Charlie spoke about the soil aggregates. These are the natural clumps formed by particles in the soil. They play a key role in maintaining moisture. In the no-till systems, these aggregates remain intact, creating pores that help soil retain water, enable better root production. This can have significant consequences for irrigation, especially in regions where water conservation is a big priority.
Yeah.
So for growers, this data underscores the value of adopting no-till practices. While it's not one size kind of fits all, these water conservation with becoming increasingly critical issue, the ability to reduce irrigation while maintaining healthy soil is a significant advantage.
Absolutely. One thing that kind of jumped out at me in terms of just looking at this graph is the different fall patterns, so sort of the different angle. You can see here, right around May 5th, there's an irrigation, and all of the soils got irrigated. This one down in the bottom, again, as Jay pointed out, this is a really sandy soil, very low vigor, very low amounts of soil organic matter. You can see it drops off really quick, so much so that they actually have to re-irrigate it. They re-irrigate, but then again, it just drops off super, super quickly, that lowest line. The full till, same thing. There's the fall pattern is very quick. It kind of crosses for a second with the every other row till, but then it drops off and has, the least ability to hold on to that water. Um, the every other row till got nice and wet, almost all the way up to saturation, and then drops off. But you can see nice, steady kind of slow dropping pattern there for the no-till. And again, no big surprise there, right? We know that the structure of that no-till block is going to be the best. And so it has that ability to hold on to water. Really important, as you're pointing out, Dana, especially in the West. Jay, do you have any other thoughts on this one?
Nope.
All right.
Great. And then, so this is the same vineyard, and we are just looking at the soil temperature from the full till versus the no-till. So we're looking at comparing two different blocks here. So soil temperature plays just a critical role in determining the rate of evaporation and microbial activity in the soil. Um, so when the top 6 to 8 inches of the soil goes above 100 degrees, we lose 85% of that moisture just through evaporation. The full till blocks experience soil temperatures over 100 degrees for extended periods. By contrast, the no-till practices buffered soil temperature fluctuations, keeping temperatures lower and stable. The data from the trial showed that the full till resulted in 69 days were exceeded 85 degrees, while the no-till above 85, it was only 29 days.
Yeah, and I
Um,
think, it's really interesting to look at that because we're all very proud of ourselves in regenerative agriculture and think we're doing the best we can to steward soil health. And really, what drives the most impact on soil respiration, we see it over and over again, is just temperature and moisture. And tillage is going to be necessary to some degree for agricultural practices. We'd all love to reduce it, but when you're doing redevelopment or you're planting, you need a seed bed. There's going to be some amount of soil work involved in agriculture forever. So I think, these slides really clearly show the buffering effect that leaving your soils undisturbed can have, both in temperature and moisture. And that really allows for the Goldilocks zone to occur for your soil biology. Right? We talk about this all the time. It needs to be just right for the soil microbes. And we'll go into this a little bit later in terms of the trade-off between temperature and moisture. But those are the two things you really want to keep sort of present in the soil while you're trying to build biology and have good microbial cycling, good carbon cycling. And they talk about that Goldilocks zone being between 65 and 85. When you get over 85, you lose a lot of available moisture. When you get over 95, 100 degrees, you start to see die off of microbes. So we like to talk about this with our growers a lot, is the Goldilocks zone, maintaining moisture and temperature whenever you can, whenever you're trying to build biology. Sometimes it's not possible in the middle of the summer if you don't have water or it's just too hot. But we know the tillage impact, both in terms of structure of the soil and in terms of just creating sort of hostile environment where microbes can't thrive.
Absolutely. I really appreciate you presencing that, Jay. I had a mentor for a long time who I worked with who was this incredible compost kind of wizard. And he always said, biology buffers all. And it's so, so true. So, if we have a lot of biology in our soils, it can buffer temperature, it'll buffer water, it'll keep us right where we want to be, right? Not too wet, not too dry, not too hot, not too cool. And we see that in our data time and time again. Biology can even buffer pH, so it really is kind of the cure-all. I really think that any type of kind of degraded or damaged soil that you have, if you add a lot of diverse biology or you cultivate the biology that's already there and get more of it active and get more of it functioning, it'll buffer all of your problems, buffer your problems away. And I think that's more and more important in California and really across the United States, where these climate extremes are more and more the norm. These huge heat waves, these unseasonable, really like late frosts, um, all of those challenges, also pest pressure, all these things. Biology is the answer to a lot of these problems. It's not going to like solve the problem like some kind of, where you just magic wave the magic wand, but it'll buffer it. It'll allow you to stay in that happy middle zone more. And so, this data shows exactly that, right? With the full till just clearly getting way hotter on the hot days, but also way cooler on the cold days. So the no-till just shows that by keeping the soil covered and by keeping more biology alive and active in the soil, you're buffering and you're staying in that Goldilocks zone.
Yeah, even the warmer days and the night and at night with the no-till, it's a pretty impressive.
Yeah, absolutely.
Yeah,
So,
we see the same thing over and over again. This is a trial we're doing with Donham, and we just recently installed here in the early fall, right after harvest. They did some tillage, and you can see it really clearly, right? Just in the first few weeks that we installed and following the tillage. The blue line is the tilled block, the orange line is the no-till block, and you're seeing those fluctuations. And this is during the fall when things are relatively mild, right? Imagine the pronounced sort of differences as we get into more extreme conditions. So again, I don't think it's, you know, anyone deserves a slap on the wrist for tilling when necessary, but we should just think about it, especially if you're trying to build up biology and switch to a more sort of holistic farming system and relying on nitrogen that's in the air, relying on more organic inputs, we're relying on the sort of biological cycling processes. You really want to try and buffer temperature, buffer moisture, and the easiest way to do that is to be smart about a tillage.
mhm.
And we see this time and time again as well in the canopy. So it's not just soil temperatures that are buffered by this, but that reflective heat that effect essentially shining a mirror on your canopy, a mirror on your plants. When you go through and you till, it just absorbs that heat and reflects it back all that more intensely. We also see less humidity, typically, less evaporative cooling happening. Um, and this directly affects crop quality. It directly affects photosynthetic processes, right? We know when things get too hot, stomata shut down, plants aren't as productive. When it gets really hot, we see desiccation of fruit. And a lot of issues come with extreme heat. So, it's not just making your soils cooler, it's making your entire farm cooler when you keep that soil protected and you keep those covers alive as long as you can.
Yeah, I love that. And I think that taking it one step further, it not only keeps your entire farm cooler, but if you could get all your neighbors to do that, then it's literally keeping your bioregion cooler. Like in my view, how we're actually going to like fix the climate crisis is local climate repair. And there's some really cool work being done, in Napa Valley right now around that. I know Todd and his Abuelitos Foundation is working on exactly that. But yeah, if you and all your neighbors or kind of an entire landscape can keep soil cool, then not only are you going to keep your individual crop and your farm cool, but collectively you'll keep your bioregion cooler.
Yeah. And shouts out to Donham too, and Tony over there. They're I think, we could talk about better farming, less inputs. We can talk about carbon cycling, but I think what will really drive this movement forward is quality, right? And we did a webinar with Dan Kittredge, Bionutrient Institute, that talked about nutrient density, and that sort of applies across the spectrum of raw products, and even processed foods if they're processed correctly, in terms of soil health, tying directly to nutrition. But in wine, we think that there's a really clear tie between soil health and wine complexity and those sort of compounds that show up pre-ferment. And Tony's looking into that. They're actually going to do some blind tasting and looking at, those links between the regenerative practices they're doing and wine quality. And we think that this temperature buffering in wine has a really clear link to those secondary compounds, those flavonoids, those phenolic compounds that make for a full-bodied and complex wine.
especially in a year where you have a crazy heat wave. Like this year was pretty mild, right? So I'm sure there's a connection there, but kind of jumping back, thinking about 2022 and that massive heat dome, the difference there would be the difference between like major dehydration and desiccation of fruit versus, oh, it was just really hot, but my plant was still functioning because I was able to buffer those temperatures and keep them cooler. Okay. So we've been talking a lot about tillage. Now we're going to talk about different termination styles and different termination sort of approaches for cover crop. So Joseph Phelps is experimenting with having, planting cover crop and then two different termination strategies using herbicide versus using grazing. Here, the blue line is where sheep were brought in to terminate that cover crop, whereas the orange line was herbicide. I think that it's really important. This might get me into trouble. I'm just going to call it out. Herbicide use doesn't just stop at killing the plants. There really is a bit of an ecocide effect of it. We've seen this, I think there's a few different kind of functions of this, or a few different ways that this is happening. But when you go through and you spray herbicide, you're not just killing the plants on the surface of the soil. There is some residual, microbial killing effects that sink into the soil. On top of that, there's a totally different sort of ecological cascade that you're setting off. When you bring in sheep, the sheep are eating the cover crop, they're pulling on the plant, and that actually triggers this enzymatic and hormonal response of the plant. Also, there's enzymes in the sheep's saliva that actually trigger it to slough off a ton of exudates. We'll talk a lot about root exudates kind of today. Exudates are essentially energy in liquid form. They're carbohydrates, they're simple sugars, they're amino acids and organic acids. Sometimes they're even more complex molecules like proteins. But essentially, it's sugar, right? It's energy that plants are releasing to the soil microbiome in exchange for nutrients. And in this case, the plant is getting eaten by the sheep, and the plant says, "Oh God, I just got eaten. I need to like regrow." So it sloughs off all this energy to the microbiome. The microbiome receives that energy and then brings a bunch of nutrients, goes out, mines the nutrients, brings it to the root, and then the plant is allowed to regrow. Um, that obviously is going to have a huge stimulating effect on the soil microbiome, and that's what we see here. So the sheep were brought in, in February, and some of these big sustained spikes are a result of that. We're seeing that in action, right? We're seeing this huge exudate flush and then the response from the microbiome, whereas in the herbicide, it's staying much lower. And we can see there's, definitely some similar shapes. So you can see here, like right after February 2nd, there's a spike in both. That's likely because there was a rain event. Same thing here, March 10th-ish, there's a spike in both. But yeah, clear winner in terms of what is doing more for your soil health. Clearly, the sheep are the way to go here. And at Joseph Phelps and at a variety of our sites, we see this kind of really interesting natural pattern of soil respiration where there's a big spike in spring and then it dies off, and microbial activity and respiration really mellow out during the summer, and then there's another spike in fall. And that's a very natural pattern. It's not a bad thing. The world is a nuanced place, right? And we want to see more respiration, but not necessarily just like tons of respiration all the time. In the Mediterranean climate, it's very natural. That's what the native biology wants to do. There wants to be this big spike of activity in the spring. It wants to kind of go dormant in the summer as it gets really hot and really dry. And then when it gets moist again in the fall and we're starting to see some rains, then the activity bursts again. So that's what we saw at Joseph Phelps. We saw everything, a big spike in the spring, everything declined in the summer, and this is the fall spike again. So what's interesting here is kind of towards the end of the summer, we actually saw the herbicide block have slightly higher respiration, and you can see that here. The native biology really went dormant, and there's still activity, but very, very little until we got that first rain. And then when we had that first rain, even just like, a half inch or so is enough to incorporate a lot of that manure into the soil and kind of reactivate that soil microbiome. And as you can see, just poof, goes way up, and then it has a very sustained kind of high level of activity. So,
Yeah.
Jay or Dana, any thoughts on this one?
No, I think it's interesting. Like you talked about it, Charlie, in the spring, you see that effect of sheep keeping the vegetation or keeping the covers in a vegetative state, right? And pushing exudates into the ground and building biology. And then in the fall, basically what we're looking at, the sheep aren't there. They haven't been there for the entire growing season. But there's still this like much more pronounced recovery of the soils and recovery of those microbes. And we know that microbes go dormant, right? They form spores. They kind of put a shell around themselves and hang out until those conditions are ideal again. And we see this over and over again. I would say with our growers who are, I hate to put everyone on like, a spectrum, but the people we know are doing an amazing job of building native biology and of managing, orchards, vineyards, farms regeneratively for a very long time in California. We see this very clear, super active period in the spring, super active period in the fall. So we always counsel, the growers we work with, if you're doing amendments or you're doing anything to drive fertility, yeah, you can do it in the summer and you might see some incremental gains, but think about when the biology is most active anyway, and really focus on those areas. Focus on building biology in the spring and focus on driving that recovery in the fall. That's going to have the it's not going to it's not going to have a sort of asynchronous effect with nature, right? Because again, what I said earlier, we can all be very proud of ourselves as growers, but we don't have nearly the effect on soils that the climate and weather does. So just work within that framework.
I love that. You're so spot on. I think that in some ways, there's all this debate around what is the definition of regenerative agriculture. And in some ways, the way that I think about it is like we find these natural waves in nature, and regenerative is to replicate that and to accentuate it. So we make that wave even greater, right? But we don't try to push a wave where there's no wave. So if we see there's this natural trough in microbial activity in the summer, it probably doesn't make sense to do a big microbial inoculant or like try to get the microbial activity to be super high because you're fighting uphill, right? The conditions, both soil moisture and soil temperature, just aren't conducive for that. So yeah, I really love that call out, Jay. This
Yeah.
is looking at just the cumulative respiration in this trial over the year. Just I like this because it just really shows that, bringing in the sheep as opposed to the herbicide about double the amount of total microbial activity throughout the entire year. So again, most of that happened in the spring and in the fall, but a really, really important, important metric to look at.
Yeah, and Dr. Rick Haney really, when he did this research, he wanted respiration to be a vector for fertility management, right? And we talk about that. You see it in the research, the link between nitrogen mineralization, carbon mineralization, and nutrient cycling and overall activity. So you can read this and basically say you created twice as much fertility with that practice, right? Because the more microbiomass, the more biological biomass there is, and the more active it is, the more cycling is happening of all these nutrients and nitrogen and carbon.
Yep, absolutely. And also, to circle back on something you said earlier, a really interesting thing about sheep. So, herbicide, obviously, you're just killing the plant. With sheep, you're bringing it in and you're actually keeping it in that vegetative state. So, instead of it going to flower, so plants, like what their exudate patterns look like is when they're in vegetative state and they're really trying to grow a lot, they put out a lot of exudates. Once they flip into reproductive state, they can't share any of that energy. They don't really have excess sugars or energy to share. They're trying to actually hoard all the energy they can to put into their seeds in order to, reproduce and spread their seed. And so by bringing in the sheep, it triggers this like phenological sort of a response of the plant, keeping it in vegetative state and keeping it feeding the microbiome.
And it's interesting because we just sort of spent some time before these slides talking about tillage impact, but I'm remembering a conversation that John Kemp had that he highlighted that he had talked to Dr. Christine Jones about. I think it was an event that Green Cover put on where she was talking about this, but she was actually saying that tillage can have a lesser effect on soil biology than herbicide application.
Yeah.
So again, I want to take all of this together and say we're not saying don't till. We're not saying you need sheep in your vineyard. That could be cost prohibitive for a lot of people. But just trying to paint a picture of all these things that can impact biology and why that matters. And again, harping on it here, there's really nothing we can do that's going to match the climate effect on biology here. And you see that early spring up until about April 30th, biological activity trends exactly with the soil temperature here, right? They mirror each other exactly. And in California, we kind of see this over and over again in the trials that we run and where we're installed, is that in the spring, the primary driver of biologic function and activity is soil temperature up to, the late spring and early summer, where it becomes moisture driven. So at some point, when things get hot and dry down, now it's not really a temperature effect. It's a moisture effect on the microbial populations. So you can think about you can't control the weather. You can maybe control moisture with irrigation, but as soils are warmer and more buffered in the spring, they're generally going to be more active. And as the summer goes on, the more moisture you can keep in the soil, the longer you can prolong that active period in the spring. So this is at that same site, and you can see April going into May. Now all of a sudden, those biological bursts, those bursts of respiration and CO2 coming out of the soil line up really, really well with irrigation events. Um, and maybe they're they're not right on top of each other like that graph before until the temperature starts to stabilize and things start to sort of moisten again in the fall. But you can see that those spikes correlate exactly with irrigation events. So the learning here, right, is as your soils warm up in the spring, take advantage of that period, build fertility, and then think about moisture as things start to get warmer, and doing everything you can to protect the moisture in the soil. That's going to allow that sort of microbial active period to extend into the late spring, and build a lot of fertility in the ground.
Yeah, absolutely. I thought these two graphs are just so fascinating because, like you said, there's this hinge point that's so clear, right? In the here, March 15th through about kind of mid-April, it's so clear that soil temperature is the limiting factor on microbial activity. And then all of a sudden, the soil temperature keeps going up and up, but we see this drop off. And then we go to this next graph, we see, oh, now it's actually about moisture, and moisture is the limiting activity are the limiting factor on microbial activity. And we still see some good spikes here. There's still definitely some good activity, but as we get later and later into the summer, even when they're irrigating, you can see it drops off so much quicker. Whereas here, they do a little irrigation. It takes a long time for the soil moisture to drop off. Here in the middle of July and August, they irrigate and it gets super wet, but it just gets pulled right through the soil profile, and they're really not seeing like sustained. It's hard to keep the soil super moist. As you pointed out, or as we've been pointing out a few times, cover crop, great way to do that. Increasing soil organic matter, great way to do that. Improving soil structure, great way to do that. Helps the soil actually hold on to that moisture. And
Yeah.
then this grower had this great insight where he did this huge irrigation after he harvested, right? September 16th or so, he said. And we saw this massive spike of respiration. And he's like, oh, that's awesome. And he said, oh, I wish I had kept my eye closer on this data because I would have done another irrigation here, October 10th or 11th in order to get a second big spike. Because again, soil respiration is sort of a vector for nutrient cycling and nutrient availability for the crop. So if you really want to put your vines to sleep for the year with a ton of nutrition, you want to keep your respiration as high as possible after harvest until you really enter that dormancy phase.
Yeah.
Yeah, the
Really.
ability to overlay two metrics all with soil respiration creates so many insights and so many aha moments for growers. It's been really fun to be a part of and to see that in real time.
Yeah,
Yeah.
we were talking to one of our scientific advisors, Chuck at Understanding Ag a few weeks ago, and he had mentioned this being sort of a critical insight, right? Is when you're doing fertility management, when you're going out and applying inputs, make sure your moisture and temperature are favorable to those inputs being cycled in the system, especially if they're organic, right? Especially if they require that microbial digestion and processing to make a plant available. So yeah, keep an eye on temperature and moisture in the spring and fall, and really try to dial in your fertility around those metrics. That's sort of the learning here.
And just a quick kind of shout out or highlight. We have a beta feature in the grower portal where we actually look at nitrogen mineralization rates. This is again based on the work that Rick Haney did, but he showed that the total amount of microbial activity and the mineralization of nitrogen, especially organic nitrogen, which is in these systems, the majority of it is organic. In that, it's coming from probably organic fertilizer, but more importantly, it's coming from the soil organic matter there. But yeah, we do have that in the grower portal. And so you can actually look at how how through improving your microbial activity and your respiration, you're actually also improving your nitrogen mineralization rates. And you can really keep an eye on that exact metric.
Yeah. And sort of carrying that thread forward, right? This is at that same site in Napa. They have one block where they're fighting vigor all the time, right? And this is sort of something that's unique to uh, growing grapes for wine. You'd think maybe as an outsider, oh, I want the most vigorous, productive plants all the time. Well, it's not actually true. You want uh, wine grapes to experience some stress. You don't want too much yield. You want to concentrate flavor. You want to concentrate quality. So oftentimes, if you have too much vigor, wine growers will go in and they'll prune and they'll try to sort of address that overproduction. And we saw that reflected where they were fighting this vigor. We saw soil respiration that was off the charts throughout the season, right? So in certain cases, you could make management decisions the other way, right? If you're having too much vigor and you're seeing way too much nutrient cycling, this is probably because there's a ton of nitrogen pumping in that soil. There's a ton of nutrients available to the vine, so they just want to stay vegetative all the time and push new growth. Um, and this really clearly to us sort of illustrated that example of if you're trying to control for that outcome and put as much vegetative growth and put as much yield out as possible, soil respiration can be that metric that you manage fertility around, or an additional metric that you manage fertility around. And if you're trying to maybe knock back some of that vigor, some of that cycling, maybe you irrigate a little bit less, or maybe you open up the field, or you pull
At
it
least,
back,
yeah, at least don't plant legumes in your cover crop, right? You know.
or apply compost if it's looking like this and you're trying to pull back on vigor.
And so on
Um,
this one, it was the green line, correct? That was the block with just insane vigor.
Yep, exactly. So this was a sort of an interesting, probably the only case we had where we were actually creating too much fertility and too much vigor, and it showed up in the respiration.
Yep. And then I believe I remember kind of the orange and the red lines here were blocks where he was really happy with yield. It was kind of like middle of the road, good. Whereas that blue line, he actually was like, I could use a little more. And again, it shows shows in the respiration really clearly with this blue line kind of almost going completely down to zero in the middle of the summer. And then, spikes a little bit in August with an irrigation. But yeah, overall, the green line was just, far and away the highest respiration, highest microbial activity, too much nitrogen available, too robust of nutrient cycling happening.
Yeah, I think this also highlights too, at least in wine grapes, right? What we might be trying to control for is an active spring, a dormant summer, and an active fall. You see that in the dark red and orange sort of trend line, whereas green is super active through the entire growing season. And that might be a good thing for another crop type. But for wine, I think we're seeing this pattern of active spring, dormant summer, active fall.
Yeah. So, kind of continuing on the theme of how to modulate microbial activity, obviously compost tea is a great way. It's kind of the before the advent of all these bug in the jug, as they like to call them, isolated microbial products, which, by the way, we'll talk more about those in a second. But this was a really cool compost tea trial. Dana, you want to tell us about some of the results from it?
Yes. So the C2 line, which is the pink line, was the block that received compost tea. I believe it was foliar fed, and then the blue line is our control line. So we're showing soil respiration in these two blocks. You can see that soil respiration increased significantly in the blocks treated with the compost tea, indicating a boost in microbial production and activity. The stimulating effects were directly into the soil and applied through just foliar feeding.
Mm-hmm. And
The
again, just the timing of it, I thought was so interesting, right? Again, looking at, it's March 16th through about kind of
Mhm.
mid to late April. That's where you're really getting the biggest spikes. So again, in terms of like insights and takeaways for me, in terms of what we learned in 2024 that we want to push in 2025, you really, if you are going to do a product, a compost tea, compost application, whatever you're doing to try to boost microbial activity, you want to time that alongside the natural burst of activity in spring.
Yeah, and this was at Gergich where they've been farming regeneratively and biodynamically for many years. And so you see that decline in soil respiration as the microbes go dormant, starting in May and June and eventually plateauing kind of into July. And so, well, we see that in these growers that are just doing everything right. And so we're realizing that's a very healthy soil microbiome.
Yeah.
And I think in
Hey,
this case, Bernat actually maybe had stopped like early April. So again, just goes to show, um, do your fertility in the spring and it'll continue to build, right? That's that inoculating effect. It wasn't like they kept it up, the entire period we're seeing that burst. I think it was like two applications.
One or two applications, yeah.
Mm.
yeah. And again, Gurgitch Hills is a great example of a place where we can say with a good amount of confidence that there's a lot of native biology that's been built up. This isn't like biology that has been brought in from the outside. This is, very much like the native biology to that area. And we see it, we were scratching our heads for a little bit because in the middle of the summer, respiration was actually pretty low compared to some of the other customers we've seen. But again, it makes so much sense that native biology in places like Napa and places in California where it really is that Mediterranean climate, huge burst of activity goes dormant in the summer as it should, and then again in fall, another big wave of activity.
Yes, and here we have the running total accumulation of the soil respiration with the blue line, the blue bars going up, indicating the blocks treated with compost tea, and then the maroon bars on the bottom are the control. And then we have the blue and red lines running across the graph, indicating soil temperature. So again, when we have that kind of that Goldilocks area of good soil temperature and soil moisture, you see the increased amount of respiration. So we're just showing the basically the relationship between soil temperature and respiration. Um, so by applying those biologicals in the spring and fall when soil moisture and temperature are at that ideal place, you're getting the most return for your investment.
Awesome. Thanks, Dana. So yeah, just cruising right along because we're already at 10:20, at least my time. This was looking at a cover crop trial. This was a grower kind of in the Central Coast region, and he was looking at different types of cover crop seed mixes. We were asked not to share too many details of this one because it'll actually will be part of a publication down the road. But we can share that Merced rye was this green line at the top with the most microbial activity. This is a really interesting one, right? Because all the lines are really moving in tandem. Sometimes when we have a sensor in one block and we have a sensor in another block, you can't really see the pattern and or sort of everything. There's a lot of noise in the data set and it's kind of hard to see. But you can see here, this is a strip trial. So these are all right next to each other. And you can really see how the soil microbiome is all moving in tandem. But rye has the highest amount of microbial activity, and then a legume mix had the lowest amount of activity. The control and the legume mix are this pink line and the blue line that are the lowest ones. And again, Merced rye has the highest. And yeah, we might ask, why is that? My working theory is rye is known for being an incredibly productive workhorse in the cover crop world. And most of that has to do with its rooting structure. A single rye plant has 13 million root branches. It has 390 miles of roots. This is, I think, the max that's ever been recorded, but still almost 400 miles of roots. And check this out, 7,000 miles of rootlets, of root hairs on a single rye plant. And where is that exudation happening? It's happening at the tips of those roots. So you can imagine if a single root or a single rye plant has nearly 7,000 miles of root hairs, that's a tremendous amount. I think I looked it up and it was like 400 square meters of surface area for a single rye plant of places where there can be an exchange between the microbiome, nutrients being brought in by the microbiome, and those exudates being released by those roots. So huge amounts of roots, really important for two reasons. One, it's a huge amount of organic matter, right? It's like those roots will slough off and die continuously, and it's actually adding organic matter very deep down into the profile. And then number two is also that's a massive amount of surface area for exchange of exudates for nutrition. So no big surprise. Or I mean, I wasn't super surprised, but it's great to see rye coming in. Overall, we saw in our data time and time again, I do think it's important to emphasize this, diverse cover crops are where it's at. Cover crops, each cover crop and each type species of cover crop provides a different root buffet, right? It provides different types of compounds. Some provide more amino acids and organic acids, whereas, like for example, a leguminous crop that has rhizobium will actually release believe it's like a flavonoid, and that actually attracts the rhizobium bacteria to fix nitrogen. And this was an interesting study I found just on the right. It's this biplot. I'm not the best at reading these, but what you can see here is basically these different cover crop varietals have different functional trajectories, right? So it's not just about a cover crop having different levels of microbial activity that it produces, but it's different types of microbial activity. It's different trajectories in terms of the metabolism and like the functional cascade that would happen based on a single cover crop. So that's why you want a diversity, because then you're feeding the soil microbiome these different root buffets at different times. And the microbiome has sort of everything it could ever want. It's got salad, it's got steak, it's got potatoes, all from these different cover crops, and also all at different times. That's another really key piece, right? In California, a lot of our cover crops will probably die in the summer, but if we do have native grasses in there, they're going to still be a little bit more likely to survive for a longer time and be able to feed the soil microbiome. Any other thoughts on this, Jay or Dana, in terms of like the different functional trajectories or different sort of expressions of microbiome?
No, I think
Yeah, I love. Oh, sorry. Go ahead, Jay.
I was just gonna say this is the first study we've done where we're looking at different like varieties of cover crop and how it's interacting with the microbiome. And it's an area I really hope our customers explore more of. I think thinking about the right mixes, thinking about how you're trying to drive the biology in the direction you want it to go, thinking about seasonality, right? And then also, we know that there's a good amount of inoculant that comes in with a seed, right? There's a lot of biology living in a seed. So thinking about where you're sourcing seed and, you know, two rye sort of two rye seeds might not perform the same depending on how it was grown.
Yeah.
And so I think there's a whole area of study and really area of fertility management that can be opened up. And we have the pleasure of working with a lot of those folks who are doing a ton of good work around cover crops. So excited to see more here.
It's so true. I just want to like double click and emphasize on that. Not all cover crop seeds are created equal, and that there's actually a ton of biology that comes inside of a seed. This is like kind of newish science, and not a lot of people know that, but you're not just getting a seed that's totally sterile. There's literally a microbiome from that soil comes inside of the DNA of that seed. It's an incredible thing. If you haven't ever heard of this, look up like rhizophagy and the rhizophagy cycle. But yeah, Dana, any other thoughts on this?
Yeah, I was just going to say I love this infographic with the root buffet showing the different types of microbes,
Yeah.
for the different types of cover crop.
Yeah, I love that they're sitting at their
That's
little
really cool.
cafeteria seats, eating their amino acids or whatever they are. Cool. So let's talk about this cool biologic trial we did at Vino Farms.
Yeah, absolutely. Can't share like a ton of information about the products just because we don't want to share too much beta, right? But this was an interesting trial where Vino was looking at a bunch of different products and doing a strip trial, putting them in the ground and seeing the effects of the treatments, throughout the growing season. And in this case, they actually didn't choose like the top performer in terms of soil activity and microbial response. They chose the second best performer because it wasn't that much behind and it was significantly less expensive. And they saw the best response in the vine from it. I think all growers, all innovative growers are doing a lot of trialing on their own, and typically they're relying on their eyes, they're relying on yield, um, using this technology or doing like Haney or Solvita analysis, right? Gives you just another vector, and that can be if you're trying to build soil biology and fertility that way, that can be a really important thing. It can also go the other effect, right? If you're using like a biocontrol product or a bioherbicide, or you're trying to control for a pathogen or a pest or disease, we've had customers run trials where they're trialing different products that are targeted at the same thing, but they want to find that one that is sort of softest on the ground, right? And has the least biocide effect on soil biology. Um, so yeah, this is another really interesting use case. And I think, before you put a bunch of stuff out into any farm system, you really want to trial. You really want to understand the different aspects of how it's playing within the ecosystem. Because you're probably not going to see a dramatic yield impact on the first year. But you might see a dramatic impact in water holding, in biological activity, and that's really exciting. And what gets our growers excited about working with us is like we can show those early impacts that we know are going to lead to changes in growing conditions down the road. So yeah, in this case, if you go to the next slide, Charlie, we saw differences in microbial respiration. We also saw differences in water holding. And this was all sort of just within the first year of the trial. They're continuing it, and we'll see sort of how it plays out. But these are the areas we typically see the first sort of impact of inoculation and improved soil health is enhanced respiration and enhanced water holding. Sometimes we see temperature effects as well, but these are the early indicators in the system.
Absolutely. And again, just to be clear, we're not saying that soil respiration should be the end all be all metric that you make all the decisions off of. That would be very foolhardy. However, we are saying it's a really useful tool to have in the toolkit. And I think it's actually quite silly to not have that. Haney tests are great. Solvita tests are great. They are just a single point in time. And as you can see from these graphs, biology varies a ton throughout the year. It varies a ton throughout the field. So having this continuous data stream is really an important one. But yeah, we pair this with soil tests where we look at a full Haney analysis. You could do a PLFA, you could do sort of a full metagenomic analysis. Obviously, we're going to be looking at yields. Obviously, we're going to be looking at crop quality. We're going to be looking at pest and disease resistance. And sort of by gathering all of these different data points, we can make a more informed decision. And that's really the ROI of agrology, right? That's really like the value that we deliver is more data to make better decisions to hopefully make more money and grow a better crop.
Yep.
So this is a case study and some data that we've shown quite a bit. So apologies if this is a repeat for anyone. I'll move through this somewhat quickly. The real interesting learning on this one was we keep having this question brought to us of, okay, I get that I see microbial activity in this one spot, but how representative of this is this of the larger block? Do I have to put out 2 or 3 or 4 of these? Like, are they going to be totally different? And so in this is a really cool trial that we're doing with Kane Thompson down at O'Neill Vintners and Distillers in Paso. And they're doing a side-by-side of regenerative organic production next to conventional viticulture. And in this graph, the purple and the green lines are the regenerative organic. So you can see in general, they're really, really tracking together. And then the orange line is the conventional. You can see flat line, way less. Again, this is looking at respiration, way less microbial activity in the conventional. We have a whole case study about this that we can share out. But the main difference between these two, which is really interesting, is we see this big spike every single day at the beginning of the day, this green spike. And the difference is we put the green one very close to the vine. And so that's actually picking up on a huge exudate flush that's happening at the beginning of the day. As photosynthesis ramps up and starts, the vine actually releases a huge amount of exudates to signal the microbiome, hey, ding, 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 like let's start the day, start bringing me nutrients. I'm going to start growing, let's like starts the factory essentially. Whereas the purple one was in vine row, but very far away from, like right in between the vines. So the farthest away we could get it from the vines. So we don't see that big exudate flush. We were actually just talking with a UC Davis researcher yesterday, and she said, this makes total sense. She's like, I'm not surprised to see this at all. This is looking at canopy absorption. So another big question we get is, okay, I get that more microbial activity is good, but how does that relate to carbon sequestration? How does that relate to soil organic matter? Can't I just track, SOM, soil organic matter, and that would kind of be the same thing as tracking microbial activity? And so this is kind of the first or part of the answer to that question, which is this is looking at canopy absorption. This is looking at how much CO2 is absorbed by photosynthesis. The blue line is the concentrations in the canopy of the conventional. Green line is the regenerative. So we're seeing lower concentrations in the regenerative block here, right? That is showing that it's actually absorbing more out of the atmosphere. If they were equal, they would be pulling the same amount out of the atmosphere. But the regenerative block is much more vigorous. There's more leaves, there's larger canopy, and that's creating more photosynthesis. That's more solar panels pulling in carbon dioxide from the atmosphere. And as we know, as we've been talking about, a lot of that energy is going down into the soil microbiome. That's what's producing the higher respiration rates, or one of the things that's contributing to it. And how does this all tie into to organic matter and to total organic carbon? You can see kind of how this plays out here. So this is a test from this year. This is year 4 in this trial. So this is the accumulation of 4 years of really good work, regenerative versus conventional. And you can see for organic matter, we're at 2.4%, whereas in the conventional, we're only at 1.4%. Total organic carbon, nearly double that of the conventional, 0.86 versus 1.51. Total organic nitrogen, also much higher. The Haney Soil Respiration Index, 30 parts per million versus 17 parts per million. Looking at the total change in the soil microbial biomass carbon. So this is the total amount of carbon in the necromance, in the soil microbial, basically the bodies. We saw this increase 9.1% from August 23 to August 24, whereas in the control block, it actually decreased a little bit. And then taking it all the way to carbon, right? That's a really important part of regenerative agriculture. For us, we always like to start with the operational benefits of regenerative agriculture, but there are really clear climate benefits as well. We need to get more CO2 out of the atmosphere, and regenerative agriculture is a great way to do it. We're seeing 12 tons of additional carbon in the regenerative block versus the conventional. And how does this translate to water holding capacity? That's roughly 20,000 gallons per foot per acre of additional water holding capacity. So in a place like Paso Robles, where they're very dry and very short on water, that's huge. That's a huge operational benefit. That's 20,000 gallons per acre that they can save.
Yeah, and I think this all goes to show, right, that it's not that carbon is a cycle, right? And if you're just measuring the output and throwing practices at the wall, you're probably missing some benefits, some operations to put more carbon in the ground and honestly create better growing conditions along the way. So, working with Agrology, you see that entire carbon cycle moving, and we're actually looking at the liquid carbon pathway in action, right? So seeing those early indicators and intervening early will lead to a better result in terms of both carbon farming and just farming in general, an operational benefit that, a more biologic system creates. I'm going to drop a little reference in the chat here. I think anyone who's going down this road and thinking about a carbon project or thinking about, regenerative ag should read Dr. Christine Jones' brief on the liquid carbon pathway. And
Can you
we're
re-drop that, Jay? It's drop it to everyone because I think you just did it to the
oh,
host.
I did the
Oh, I got
panel.
it. I got it. Boom. There we go.
Yeah. So, yeah, we're just starting to sort of open a window into the liquid carbon pathway and how it can impact fertility and how it cycles with climate and practices. And yeah, really excited to further all of these trials and add a bunch more that hopefully share some really key insights with our customers in the industry at large here.
Yeah. Well, couldn't sum it up better. Thanks so much, everyone, for your time. I don't see any questions in the chat. I'm not sure. Maybe the chat isn't working. I don't know if someone can just throw in a message just to see if it is. If there are any questions, we'd love to answer them. While we're waiting for that, if anyone is interested, and we'll do a follow-up email with a recording, and Jay and I actually just put some interesting resources together in terms of the soil microbiome and exudates and sort of the dialogue between the two. Okay, cool. Looks like we did get a message in. So, yeah, if there's any questions, we're happy to answer them. You can send us an email to any of us. And okay, Pam's got a question from the ROI slide from Vino Farms.
Yeah, so those lines were the different products and a control. And that's not like ROI, that's just looking at soil respiration. And on the next one, we're looking at volumetric water content. So those are the actual products in the strip trial and how they performed compared to one another in terms of volumetric water content and water holding, implied water holding there, and soil respiration and all the implied benefits of soil respiration and microbial activity. And again, can't name the specific products, but can say they actually didn't pick the best performer in either category because it was really expensive, and there was observed benefits in the vine. They chose the second best in terms of respiration, volumetric water holding, and just because it was more cost effective and had a really good vegetative response in the vine.
We got a few more questions coming in. I think we just opened up the chat. So yeah, they're all raining in. So we have one that is kind of an interesting one that we can riff on for a while is our winery is beginning to look for regeneratively grown grapes. So that's a little bit outside of our realm of expertise, but I do have some great stats that I can share really quickly on overall growth in terms of regenerative and different sort of claims. So let me just share my screen. Let's see.
And while you're doing that, Charlie, there's a quick question on is it a good idea to till compost in or just put it on top? I think the answer is it depends, what type of compost it is and what you're going for.
And when you apply it?
You apply it. I think if there's good moisture, there's good rain, getting a top dressing, you get that inoculation effect the same as you would from like a compost tea. Um, if it's a really woody compost and you're just going for like incorporation of more carbon, maybe it does make sense to till it in,
Yeah.
But I think, yeah, look, the guidance we hear by and large around tillage is just make sure it's followed by something to cover the soil, whether that be, mulch or living cover, seeding.
Yeah,
Still leave your soils bare
totally.
after.
That's kind of arcing back to what you were talking about, that quote from Christine Jones, which is like she said she would rather see tillage that was quickly followed by a cover crop or some sort of roots in the ground, right? Living plants versus an herbicide. And that's kind of controversial. And so in row cropping in the Midwest, regenerative row cropping is no till oftentimes, but with an herbicide burn down. And she was saying that, in her opinion, you actually would have a better soil health response if you did a little bit of tillage, but then instantly kind of put living plants back in. So yeah, overall, in terms of to till or not to till, context really matters. That's like the
Yeah.
key one of the key pieces of regenerative agriculture is it's all about context. And that is, again, where agrology comes in, is we kind of help put more data and we give more data points around what your context is.
Yep. Not all tillage is created equal, clearly. Whether you're using like a power harrow to just incorporate the compost in the first 2 inches, or you're using a full-on rototiller versus a spader, there's a lot of
Mhm.
different attributes to that.
Totally, versus a reverse tiller. I'm like a huge fan of
Yeah.
those. So this is just a quick stat to kind of show some growth in sales from different label claims. We see here, organic ingredients is up 5.4%. Overall, regenerative organic ROC is up 11.7%. B Corp is up 7.1%. Glyphosate free, again, no herbicide, or at least not that type of herbicide. The most infamous one is up 20%. So we are seeing a lot of growth in the regenerative sector overall. I think we're definitely seeing that in regenerative grapes as well. I don't have any like direct stats on that. If anyone who is a guest here has any insights, feel free to chime in on the chat.
Yeah, I think there's a question. What is this slide? Is this food? Um, it's a report, I think, put out by Regenified. Is that right, Charlie?
Correct.
Yeah, so it's just kind of
No, this one wasn't actually Regenerified. I'll track this one down, Pam, and I'll send it out in the email.
and then a bunch more questions coming up. So one, how do you measure SOC sequestration with the Agrology probe? So we don't measure SOC, like the organic carbon, like TOC or SOC, with the probe. The way we approach sequestration is through net ecosystem exchange. So like similar to something like that would be measured with an eddy covariance tower. Um, and that's actually not a feature right now, but we've done our first sort of experimental model, and we have some really promising results around that. We'll be releasing it in 6 months. So actually looking at, the CO2 into and out of the soil and the atmospheric concentrations of CO2 and calculating sequestration that way, as opposed to going down the road of looking at TOC and SOC. We see a lot of innovation around soil analysis, and we think that'll be continue to be used in carbon sequestration and carbon accounting. We're hoping to bring sort of a different data set that complements it, right? So looking at sequestration from the gas phase, and the way that's been done for a very long time is with net ecosystem exchange and eddy covariance towers and looking at gas flux that way. So that's the sort of approach that we're taking around sequestration.
Yep. Just a reminder, this all is being recorded and we will share it out in an email afterwards. There's a great question from Erica. How do we partition microbial respiration from root respiration? And if the treatment also impacts both plant growth, root growth, and the microbes, then is it necessary to account for this? I think that, yeah, you pointed a little bit to the answer in the question. We don't partition those out. There's no easy way to do that in terms of the respiration that's coming from roots versus microbes. However, we know that if there are roots, they're feeding the microbes, right? So they're separating the roots and the rhizosphere is actually, I think, kind of separating two parts of one whole, right? The soil microbiome and the activity of the rhizosphere are super interlinked. And so we don't separate those out. And as you said, if there's a treatment that is going to boost both root growth, exudate flushes, and microbial activity, we don't really necessarily view it as super. I mean, it'd be great if we could partition them, but there's no way to actually do that. And since they're two parts of a single cycle, we view them together.
Yeah, and it is interesting. We have observed some like negative fluxes. So like CO2 getting absorbed back into the soil pores in the ground. We don't really know what's happening there, but our best guess is that is root respiration. And we see it more pronounced in more biologically active soils overall. So
Or root absorption, rather. But yeah. Yeah.
more roots than happening. But it's a great question. Maybe we'll have that answer eventually and be able to partition it out. But for now, we're kind of looking at respiration holistically.
Brian has a question around when tilling on the soils and these experiments, did any trials just rip the soil? I don't believe we have any that was just ripping, but we do have the Donham slides that we showed. he did an interesting thing where he used a soil pulverizer, which is actually a really crappy name for that in that implement because it doesn't actually pulverize the soil, but it essentially like pokes a bunch of holes into the soil. It doesn't churn or rip or disc the soil at all or cut it up. It just like adds a bunch of holes into the soil. And that was the no-till versus the tillage was traditional, I believe a traditional implement. Um, so no, I don't think we've looked at just ripping, and we'd really love to, Ryan. If you happen to be doing that trial, we'd love to jump in. I think yeah, like Dana said, not all tillage is created equal, and different implements have very different effects on soil structure and very different effects on microbial activity. We know that from the academic research, but we haven't been a part of the trials ourselves at this point.
Really interesting
Yeah, to
question.
clarify, oh, Tony and Donham used that just to create a seed bed for that cover crop going in. So
Yeah.
just to kind of loosen that soil up
Yep,
since they
and
were getting
incorporate
really
a
poor
little bit of
germination.
oxygen, right?
Hmm.
The question about variation of genotypes of the same species and the interaction with that genotype and a treatment. Um, yeah, that's the kind of research we would love to do more of. I don't know if we have any examples of that, Charlie.
Not
Um,
not off the top of my head.
yeah, but I think we've been out with this technology for 2 seasons. And I would say we're sort of in the early adopter phase of our company. So yeah, that hasn't been something we've looked at. We've looked more at practices than specific genotypes, but I think you could remember that slide from the different cover crop species and how they're performing um, from a sort of respiration lens. And you could run a similar trial, but with different genotypes and how they interact with specific practices.
Yeah.
That'd be a really interesting thing to look at.
We have a question from Tomas around soil moisture and soil moisture temperature from different depths where the sensors installed the same distance from the dripper, same soil type, etc. And then we have another question around soil type heterogeneity across a field. So, similar to soil samples, there always is the challenge of spatial variability in a field. We always try to install our sensors the same distance from a dripper. We always try to install our sensors the same depth, but all fields are variable, right? This is not a laboratory, and our systems are not going into systems where it's just a single factor analysis. That's what makes our data sometimes a little bit more complex and also interesting because instead of looking at, for example, how a plant responds in a laboratory setting, we're looking at how plants are responding in the real world or how the soil microbiome is responding in the real world. So yes, we always try to place them same distance from the dripper. We try to keep it in the same soil type. Like we'll look at a stratification and look at soil types across the field and make sure we're all in one soil type. And if there's a different soil type, we'll put different sensors in there. But yeah, it's always difficult in the real world to set up your strip trials to really ensure that it's an even playing field because oftentimes it's not. We actually had this really interesting example where we were doing research with a biologic company earlier this year, and they set up a strip trial. And luckily, we put in our sensors in early, and we saw, oh, this is a completely different because of the previous research that had been done on that field. One side had way more microbial activity and way better water holding capacity. So essentially, they were trying to set up like a side by side, but it was on a very skewed playing field. And so some of I forget if it was the treatment or the control ended up being on that high side where there was already a lot more activity. But luckily we were in early enough that we could actually see that and we could kind of adjust for it a little bit. But
Yeah.
yes, the real world is never an even playing field.
Yeah, and two things we're trying to do this with our software to sort of like combat that variability. One, we're trying to add some lens into like evapotranspiration, a big piece of the water cycle. The other is we're creating a machine learning model that sort of calibrates all of our moisture data across all different soil types to calculate volumetric water holding. So we gather a lot of data, and we can start to see where field capacity is, right? When we're installed in a farm for a season or two, we know when it's going to be really wet, when there's a lot of rain happening, when the soils are sort of saturated. You can also sort of tell that with soil tension and some other data metrics besides just soil moisture. So yeah, part of the way we address all of this stuff is feeding all the data into a machine learning model, and then using that to model volumetric water content and field capacity. So we're sort of like trying to compare fields to themselves, trying to flag where field capacity is, right? Where that saturation level is, and then manage soil moisture and water holding from there.
Mm-hmm.
And again, that only gets better as we deploy more.
Yeah.
Uh,
And that brings us to another point of oftentimes our customers ask, how do I stack up? Am I doing good? Like, how do I stack up against other fields? And it's a little unfair to compare a field to another field, especially when we're talking about like different regions or different soil types. So we really try to compare fields to themselves and really learn about kind of directionally where they're headed. Are you improving? How quickly are you improving? And diving in and being like, okay, let's look at these two or three different kind of programs or sets of practices and see what's going to yield the best results on in your context, in the context of this particular field or this particular, crop and field. A few other questions. Let's see. From Mohammed, he's saying, excited to see that Regen Ag practice is making a little bit of impact in the short run. Should we expect to see this kind of get added up in the larger run? And he's asking about accessing the data source or some references, and then also any comments on occasional till rotations as an alternative option to zero till. Um, yeah, so I mean, for example, in wine, we know that occasional tillage often is kind of necessary to fight compaction. And Agrology's stance on tillage and on any regenerative program is that less dogma, the better, right? Like, let's let the data speak for itself. Let's center our strategy around outcomes. If you're building soil health, if you are improving your microbial activity, if you're improving your soil structure, soil organic matter, TOC, then I think a little bit of tillage is okay if that's part of the program. Because again, we have to balance sustainability goals with production goals. You cannot be a sustainable farmer if you're going out of business. And so I always viewed farming as like a massive algorithm where you have to weigh a million factors. And really, our stance is that soil health it needs to be a very important part of that conversation and that mental dialogue around what I should do. But we don't think that like no till is the only way to go. We think that actually letting go of that dogma is a really important thing. And our data kind of allows folks to do that, right? Because we can look at and actually see, okay, look, I have to till because I'm fighting compaction in this block. We can know what's the cost going to be of that. How is that going to affect my soil health? And what are the practices that I can do to help my soil rebound really quickly after that?
yeah.
Exactly. Yeah, we work with a grower that has been certified regenerative, and they've been no-till for over 10 years, and they are considering tilling because there's a vole problem in that vineyard.
Yep.
We just had this conversation last month, I think. And what we can do is we can also monitor that soil respiration to see what it's been in the past year versus what it is going forward after that every other row-till that they're going to consider trialing.
Yep. A few other quick questions. How deep are the probes going down? The respiration is right at the surface, and typically we're in that top 8 inches of topsoil. When we're looking at respiration, we want to look at the rhizosphere, so we're looking at soil moisture in the top profile of the soil. But quite often we'll put a probe deeper down in the root zone if we're looking at grapes or any kind of perennial system. And then we have multi-depth probes available as well. So it kind of depends, and the customer, the grower, the trial uh, really have a lot more control over probe depth than we do. It's their call.
Yeah.
The other question, how do you manage fields that do not have uniform soil type? Again, when we're doing strip trials, we try to find as uniform conditions as we can. And as Charlie mentioned earlier, we try to get in before the treatment or the experiment so we can see those baseline differences and kind of baseline the trial before whatever happens that we're trying to measure the impact of. Um, so yeah, we're learning there, and I think part of it is moving the sensors around. If you're trying to get an understanding of an entire field or block or management area, you got to move the sensors around. But in a trialing setup, like we've been talking about today, strip trialing essentially, we try to find as uh, congruent conditions as we can, and we try to go in early so that we know how these ecosystems and soils are performing before we add an additional variable to measure.
Mhm. We have a question
Um,
around nitrous oxide and methane. So we currently do have the ability to measure nitrous oxide. We don't have the ability to measure methane, although we're moving in that direction and hope to have that soon. Maybe, I think within 2025 is our goal. It's a really difficult gas to measure. I know there's a huge opportunity there for us, and for really agriculture in general, right? Especially around rice production and kind of these modified flooding schedules. With nitrous oxide and organic versus synthetic fertilizers. It's a great question. It's a big conversation. We are doing some cool research around the reduction of nitrous oxide emissions based on a biological inoculant that is a nitrogen fixing microbe. And we know that organic nitrogen sources require microbial activity to mineralize, and that oftentimes that is going to be kind of a slower release or a release that's more based on a plant's schedule, right? That's the amazing thing about the dialogue that happens between roots and the microbiome is the roots are actually signaling the microbiome through hormones and through enzymes and through other kind of biochemicals. There's a communication there, and it's signaling, hey, I need nitrogen, go get me nitrogen. And then the microbes go out and then they mineralize it and they bring it back. Again, I'm just kind of simplifying it here, and I'm not a PhD, so yeah, I just know enough to be dangerous. But I do know that there is a completely different sort of mechanism around how nitrogen is being cycled there. And I think in general, we can say that we would see a reduction in nitrous oxide emissions.
Yeah, and I think we don't have enough data to sort of answer that. We're just in the beta phase with our nitrous technology. So it's available, but we have limited deployments. And where it's being used is mostly looking at like a standard ag practice and an additional product or different rotations of covers, or different rotations of practices and how that can affect nitrous oxide emissions. And what we're seeing is that emissions factors don't really fit
Yeah,
uh,
we can tell you that for sure.
outside of the systems that they were developed in, which is by and large like corn and soy rotations in the Midwest. You apply even that same system in an arid environment, and the emissions will be very different than the emissions factors would predict. When you start looking at niche rotations or products, it's even more variable from what the sort of biophysical, biogeochemical, and biophysical models would predict around nitrous volatilization and emissions. So, yeah, it's kind of a new frontier for us and Olive Ag, and we're really excited to look at that in more detail as these sensors become used and adopted more.
Totally. Yeah. On that one, Pam, I would definitely recommend you. There's a ton of scientific literature out there around how organic versus synthetic fertilizers have different nitrous emissions factors. But like Jay said, kind of our biggest takeaway from 2024 and our data this year was that the emission factors really don't cut it. Anytime you're in a specialty crop, anytime you're outside of kind of the Midwest climate, anytime there's like any sort of if it's not super mainstream in terms of like Midwest row crop, the emission factors are likely not super accurate. And so that's where there is this really great opportunity to actually measure your nitrous oxide emissions and kind of create your own emissions factor, right? If you have the ground truth data, that's a place where there's a huge opportunity to show greenhouse gas reductions and also to not be penalized for emissions that are your estimated emissions, but that aren't actually accurate. Well, with that, we're at about an hour and a half. So I think it's a good time to sign off. If you have any other questions, feel free to email any of us. Our emails are all really easy. It's just our first name at agrology.ag. I will send a follow-up email to everyone with the recording as well as some of the other resources that we referenced. And thank you all for being here, and thank you all for your great questions and engagement. Yeah, Jay or Dana, any last question or last thoughts?
No, I think yeah, if you want to talk deeper about the data, we're somewhat restricted what we can share, just because this is our customers' data, so we have to ask them permission, but we're always happy to talk sort of about ideas and research, and yeah, so just reach out if this inspires any kind of thoughts from anyone in the audience today.
Thank everyone so much.
Yeah.
You have a great
Yes.
holiday, and yeah, we will talk to you all soon.